Welcome to today's Gene and Cell Therapy lecture, where we'll focus on gene transfer systems for stem cell gene therapy. So the content of this lecture will be somatic versus gene line gene therapy, which we'll define. We'll talk about vector systems, which are currently employed, with a very special focus on retroviral vectors, gamma and lentiviral vectors, discuss the architecture, what the replication, how the replication cycle works, necessary elements we need in cis and transfer vectors, the integration patterns into the genome, and how we can improve the architecture to improve the safety of these vectors. So what is gene therapy? Gene therapy, depending on who you ask, you will hear a different definition, and also the European Medical Association, the EMA, defines it a little bit different, as you would expect, based on their subdivisions of ATMPs, and it would be a part of an ATMP, an advanced therapeutic medical product. So the most obvious, or a definition most would agree on, is that it's introduction of nucleic acids into a patient's cells for treating an inherited or an acquired disease. We differentiate between somatic gene therapy and germline gene therapy. Somatic gene therapy is, of course, deals with somatic cells where you introduce nucleic acids, and the most important part to look at here is, or to recognize, is that there's no transmission to the patient's children. That's an important hallmark. Germline gene therapy would lead to transmission to the children of the patient, or to the children as such, by introducing the nucleic acid into an egg sperm, leading to inheritance. That's something which is forbidden in most, if not all, countries. There are two basic approaches how to do gene therapy in most of the ongoing clinical trials at the moment. Gene compensation, so you have a defective gene, say, and you compensate for the function by introducing the intact or functional gene, for example, with a retroviral vector or whatever, by whatever means. Or you repair the defect if you know where it is, for example, by using CRISPR-Cas, genome editing, which would be the most elegant way how to approach it, if you have the methods there. And for many, many years, we didn't have them. So now, in future, we're going to focus more and more on doing real gene repair. Which vectors or gene tags are currently being used? In many of the trials, adenoviral vectors are being used, which don't integrate into the genome. But in most of the studies, retroviruses or lentiviruses are being used. Unfortunately, they're often separated, although they belong to the same family and mechanistically replicate and work very, very similar. So for that reason, I just introduced the slide once again to show what the family of the retroviruses. So the lentiviruses are retroviruses. Yeah? And what is commonly meant when they differentiate between, or when colleagues differentiate between retroviral and lentiviral vectors, retroviral vectors are commonly the gamma retroviral vectors shown down here, where mouse retroviruses, for example, belong to. But there's a whole bunch of different retroviral vectors, like the spumaviruses, foamy-virus, and so on, all those having common mechanisms how to replicate. Because the retroviral vectors are the most commonly used ones and very, very effective, we have to stepwise go through the replication and take a closer look on how the particles are formed to be able to construct these gene therapy vectors. So this is a very simple scheme of a retrovirus, in this case of a mouse retrovirus. What we have are these knobs on the surface of the envelope. We have a lipid bilayer with the surface glycoprotein, which belongs to the ENV gene, or encoded by the ENV gene, which binds to a receptor and enables the virus or vector later on to enter the cell. We have the surface transmembrane glycoprotein, the transmembrane part, which, as the name says, anchors the surface glycoprotein in the lipid bilayer of the virus. We have the group-specific antigens, the GAG proteins, the membrane-associated matrix protein. We have the in the mouse, for the mouse retrovirus, an icosahedral structure, which is not the same in the different retroviruses, encoding the or forming the capsid. So the capsid protein, part of GAG, and the nucleocapsid as a shell enclosing both genomes, which are two copies of an RNA of positive polarity. So a single-stranded RNA of positive polarity means that that RNA possesses messenger RNA polarity. So positive polarity is messenger RNA polarity, meaning that this genome can be directly translated. And we have an enzyme, the POL, which is an RNA-dependent DNA polymerase, or briefly reverse transcriptase, which also contains an RNase activity called RNase H and an integrase activity for integrating the reverse transcribed cDNA, complementary DNA, into the genome of the host after infection or transduction if we talk about a vector. So how is transcription controlled in a classical retroviral vector, and how is the genome organized? So in any retroviral vector, if you look at the genome organization, we'll always find GAG, POL, and ENF gene regions. It's all the same in all retroviruses. And you have flanking long terminal repeats, LTRs, which are organized by a unique 3' region here, a unique R region, which is duplicated in the genome, and a unique 5' region. And in a moment, I hope you'll understand why the U3, the unique 3' region, is at the 5' end on the left side here. Because, to be in short, reason is the replication mechanism, but you'll understand it in a second. So the U3 region contains the enhancer promoter region of the vector. And this enhancer region has many binding sites for transcription factors controlling the transcriptional activity of the retrovirus, and usually is a very strong element, and as enhancers influencing also the transcriptional activity of a whole area around the integrated retrovirus. So genes upstream, downstream, over a have long-range activity. So this slide just shows a little example of a few of the transcription factors binding to an enhancer, the U3 region, in this case hematopoietic transcription factors in part like GATA, AML, and so on. The only message here is that there's a high density of binding sites for transcription factors in retroviral enhancer regions. So how does gene expression work now? Transcription starts with the promoter at the beginning of the R region, so the +1 site, where the cap, so methylguanosine cap is put on the RNA, and from there transcription starts and goes all the way through the end of the R region of the 3' LTR, where polyadenylation happens. So this is the genomic RNA, positive strand. What happens with that? That happens in the nucleus. This is the integrated provirus, integrated virus in the genome of the host cell. Something happens with that RNA after transcription. So we have, to make it short, two types of RNA species which are formed. One is the genomic RNA, which can be directly translated then when after transport out of the nucleus into the cytoplasm. We have the 5' cap, we have our start codon, AUG, and a stop codon at the end of the POL region, UAG, and also there will be, and I'll show this to you in a second also, at the end of the GAG region, so between GAG and POL, there's also a stop codon. But to keep it simple, one polyprotein is formed, is translated from this messenger RNA containing GAG, the GAG and POL region, and the components of GAG and POL I showed you before, which are then formed by proteolytic activity. So we have a splice donor and a splice acceptor within the RNA, which is being spliced by the splicing machinery of the cell. As a consequence, the start codon of the envelope part is put close to the 5' cap. So that's how the virus gets the envelope protein or glycoprotein expressed. After splicing, getting the start codon of the envelope gene close to the cap structure. So how is the whole genome packaged? And if you would be a biomechanic or construct this thing, of course, you would think about, well, where should I place the packaging signal if I only want the full-size genome to be packaged? And the answer is quite obvious. I think so, at least. You would put the packaging signal somewhere in the area of the intron which is spliced out, so that the spliced species is not being packaged, because if you would do that as a virus, you would be dead. And the packaging signal is directly behind the splice donor site, abbreviated with a psi, or called psi. That's the packaging signal, and that's what is spliced out in the form encoding the RNA species, encoding the envelope protein, so that only the full-size RNA is being packaged into the viral particle. So now, how is the GAG-POL region organized? As I said before, and as you saw it in the viral particle, the GAG, group-specific antigen part, encodes the matrix capsid and nucleocapsid and has a stop codon. This stop codon is used in, say, 95% or about 90%, whatever, of the times, but in certain cases, the ribosome continues to translate, in about 5% of the cases, to translate the POL region. That makes sense. Evolutionary, of course, anyway, that has evolved because you need a lot of the building blocks to build up the viral particle, which is the GAG region, but you don't need as many molecules of the polymerase to be sufficient enough to replicate. So the POL region containing the protease at the N-terminus, the reverse transcriptase activity, and the integrase. In some of the retroviruses, you get a frame shift in the region where the ribosome comes to the stop codon, stalls and slips back one or two nucleotides, or goes forward one or two nucleotides and continues translation at a different frame, but not in all cases. This is a little bit different between the different retroviruses. So the ENV or ENV region contains signal recognition particle, the surface protein, and the transmembrane part. The envelope is translated into the or at the endoplasmatic reticulum and then goes through the Golgi protein to the Golgi compartment to the surface of the cell, where it's presented, and then gets together with the other components of the virus during budding. So how does replication work? Behind the LTR, behind the RNU5 region, we have a so-called primer binding site. Any polymerase needs a 3' hydroxyl group to be able to do its job, to polymerize something from 5' to 3'. So in the retroviruses, the primer is a tRNA species, usually a specific type of tRNA, which binds to the primer binding site, usually 18 nucleotides before the splice donor and after the U5 region. where then reverse transcription starts by the RNA-dependent DNA polymerase. So let's go through that. The tRNA binds, and it doesn't have, it seems, this typical clover-like clover leaf structure, you know, from the textbooks, but unfolds a little bit to be able to work as a primer and reverse transcription. So what the polymerase does, it starts synthesizing cDNA, complementary DNA, and stops there. And this short species of cDNA is called strong stop DNA. Why is it called strong stop DNA? If you do a radioactive labeling of cells which have been infected with a retrovirus, you see a very, very strong radioactive signal when you separate everything on a polyacrylamide gel. And that's this strong stop RNA, a short species which you prominently find. The RNA's H activity digests the RNA template, not completely, but to a certain extent. So now we have something which is complementary to the 3' end, to the R region there, where strands which can either occur or at least can hybridize there so that the reverse transcription process can continue. So after first strand exchange, within the molecule, within the RNA molecule, in cis or in trans, intramolecular, reverse transcription continues through the whole genome, leaving short pieces of RNA still there, which then act as primers for synthesis of the double-stranded DNA. So strand displacement synthesis happens, and you get a double-stranded piece of DNA, which is not the complete provirus yet. So synthesis has to be completed. The rest of the RNA pieces have to be degraded. That happens by RNA's H activity of the reverse transcriptase, digesting here. The primer, the tRNA. One of the questions which also arises here is why during reverse transcription not the complete tRNA is being reverse transcribed, but only 18 nucleotides. And the reason is that the 19th nucleotide is a modified base, which the reverse transcriptase cannot use. So reverse transcription halts after the 18th nucleotide because of the 19th nucleotide being modified. So what happens then? This seems a little bit complicated, but it's not really. We again have hybridization of the primer binding sites and continuing reverse transcription so that we end up with 2 complete LTRs. And that explains now how the organization is and why we have a U3 region at the 5' and the unique 3' region. So which region was copied to where? Think about it again by going through the whole replication process. The U3 region was copied to the 5' end. The U5 region from the front was copied to the 3' end. This knowledge is important, or one of the important parts, to be able to construct more safe vectors, or vectors with an improved safety profile. We'll come to that in a moment. If you want to get more into details or have another animation of the retroviral replication cycle, I can highly recommend the lecture of Vincent Raccaniello from Columbia University, a virologist, where you again can follow retroviral replication, probably a little bit more fancy than this PowerPoint slides. We're able to show it. So now let's, on the knowledge we have, build a retroviral vector. What would we need? We need components in cis, meaning, well, you want to transfer your favorite gene, and that has to be on the genome, on the RNA. and you need some signals on the same RNA because that's the species of RNA you want to get packaged into the viral particles. So you need a packaging signal, you need the primer binding site, that all that stuff has to be reverse transcribed. You have a promoter from which you would express this RNA, which has to be packaged, a poly-A tail, and so on. The gamma retroviral vectors are simply organized. You have some additional elements in cis if you use a lentiviral-based system like the ref-responsive element shown here, the RRA region, or the central polypurine trap, or flap called. So some additional elements you need for lentiviral vectors. What do you need in trans? Well, you want to build up the viral particle, and for that you need the GAG-POL region, or GAG-POL proteins, the polymerase and the group-specific antigens, and of course the envelope, which you can exchange for the envelope of other viruses. And if you use a lentiviral vector in the commonly used vector systems, the ref protein, which binds to the ref responsive elements, the RRE. So with all that knowledge, you really can build up very, very complex retroviral or lentiviral cells. Let's put the common term is retroviral vector. I'm going to use retroviral vectors which work very well. So this is just an example of a vector which is syn. So the U3 region was deleted so that during reverse transcription, the deleted part, the part not being there, is transferred to the 5' end. So the enhancer region is dead. So the LTR controlling transcription is transcriptionally inactive. It's dead. So this vector, for example, contains an internal promoter, the phosphoglycerate kinase promoter, which controls expression of a reverse transactivator, which responds itself as a protein to doxycycline. This acts as a DNA binding factor, as a transcription factor, which binds to the TET operator because it binds tetracycline or doxycycline. And by doing so, if you add doxycycline, that binds to the mineral promoter, and in this case GFP and the transcription factor are being expressed, which then can go into the nucleus and do something with the cell and label the cells green. So this is a complex vector which has been constructed on a piece of paper basically, and then of course cloned, and that works. Yeah, this is just showing some data here. When you put doxycycline on the cells, this is a flow cytometry diagram, you find a shift showing that the cells are green. So if you would look through the microscope, you would see green cells. Without doxycycline, it's not being induced, it's off. And these are just two different clones showing that. And on the bottom, not so important, showing that the biological properties of these cells, these are differentiating embryonic stem cells of the mouse, showing formation of hematopoietic cells. But that's not so important. Important is that you can build inducible, for example, complex retroviral vectors which act as you want them to act. So how can we translate all this knowledge into clinical praxis for doing gene therapy? Because that's what our goal is here. So as I have mentioned already in the lecture about stem cells, the stem cells are one of the most interesting targets for performing gene therapy, because you would, in principle at least, only have to treat the cells once, transplant them, and for the rest of the life of a patient having cure or a genetic correction. So to remind you about the hematopoietic system, we have long-term repopulating hematopoietic stem cells, LTHSCs, we have transit amplifying cells, we have multipotent progenitors, and those at the end differentiate towards all the different blood cells we have in our body, where you see if you're sick, the effect of a defect which is already at the level of the stem cell there. So for that reason, genetic correction of the hematopoietic stem cell would lead to a correction of the disease, of the functional effector cells. So our current standard, when doing stem cell transplantation, is using allogeneic hematopoietic stem and progenitor cells. And because you, with that transplant, also the immune system of a foreign donor, an allogeneic meaning in contrast to autologous, that you have a foreign, somebody else, unrelated donor who is matched. You have the problem of rejection reactions because of the immune system recognizing the recipient or the donor as being foreign. For that reason, we have to do a lot of tests before performing such a thing with a patient to avoid as good as possible rejection reactions, or only having wanted rejection reactions, which can be the case in malignancies, for example, leukemias. But if you want to avoid rejection reactions completely, the best thing would be to use the patient's own cells. That's really desirable under certain conditions. So the very first gene therapy trials which were successful were the ones trying to treat a severe combined immunodeficiency, or ICSID. And what was done is the bone marrow stem and progenitor cells of the child affected were collected, the autologous, so the patient's own cells, were collected, retroviral gene transfer of the intact gene or coding sequence was introduced into those cells, and those cells were directly given back to the patient transplanted. So what you would expect is you would have hematopoietic stem cells, you would do a transduction, so we don't say infection if we use a vector instead of not an infectious particle. So you don't have replication of a virus which just infects other cells and so on, but just transfers a gene into a cell and then nothing else happens. It's not infective. So that's the difference. So we transduce cells, and because retroviruses integrate more or less randomly into the genome, into open chromatin areas, we get different integration sites, which are depicted here by different colors. So each color represents a clone with a certain integration site. In vivo, you would expect to find those clones, if stem cells have been hit or have been transduced, in the effector cells, in the peripheral blood, all the different colors, all the different integration sites with a certain frequency. And if that polyclonal reconstitution, how we call it, everything would be fine. So in the case of ICSID, so X chromosomally linked severe combined immunodeficiency, which is the most common severe immunodeficiency, which is caused by mutation in the so-called common gamma chain. That accounts for about 50% of all SCID diseases and is associated with severe diarrhea, pneumonia, fungal infections, and so on. And you would die without treatment within the first year. So such children were kept under sterile bubbles. For that reason, those children were called bubble children, or in this case, bubble boy, like the picture here of David Feather, who unfortunately died after transplantation. So if you find the classical transplantation conditions of a matching donor, you would have about more than 90% survival rate. With a mismatched donor, so not fitting as well, the survival rates are lower, as is the case for allogeneic bone marrow transplantations. Rates increasing also because of better protocols in the meanwhile. So in the first treatments, 10 patients were treated in London, 10 were treated in Paris in the Neckar Hospital. It was a big success at the beginning. So the gamma seed, just to follow up on that and complete that, The gamma C chain is a subunit of the receptor complex, which is associated with a number of interleukins involved in development of immune cells associated with interleukins, for example, or interleukin receptors for 2, 4, 7, and so on. And if that is lacking, you can imagine that the immune system doesn't work anymore because of so many receptors dependent on the association of the gamma chain. So how did they do the treatment? They used a very, very simple gamma retroviral vector derived from the Bologna leukemia virus, having both LTRs, both full LTRs. So just to remind you, with U3, RU5 regions, and only the gamma chain complementary DNA as the coding sequence. Those vector particles which were produced were pseudotyped. Pseudotype being means that you would exchange the naturally occurring surface envelope protein for an envelope protein from a different virus. Produce those particles in so-called producer cells, cell lines, and use that for treating. So they transduce CD34 positive cells, hematopoietic stem and progenitor cell containing stem and progenitor cells. And again, here what you can see is a flow cytometry diagram or diagrams showing the success of the gene transfer. So on the x-axis you see CD34 expression because these cells were CD34 positive and enriched for that. On the y-axis, expression of the gamma chain, which is, I think, very clear, which you can clearly see over time. So it was a big success, and in 2002 ended up in a publication in a very prominent journal, the New England Journal of Medicine. But unfortunately, 1 year later, something else had to be published again in the same journal, namely adverse events which happened. So what has happened? What happened is that some of the patients developed a leukemia because of the treatment. So what is an adverse event here is that the integration of a retrovirus can, of course, also happen near an oncogene, next to a gene which controls cell cycle, the proliferation of the cell. And if that happens, what can happen as a consequence is that that cell doesn't differentiate anymore, which it would become a leukemic stem cell, which is blocked in differentiation. And that is basically what has happened in those patients. So why? Why did it happen? What is the mechanism? What we know now since many years of investigating the integration sites of lentiviruses, of gamma retroviruses, of the different retroviral species, what was found out very soon, of course, after that event has happened, is that the retroviral vectors, the gamma retroviral vectors, integrate very close to transcriptional start sites in the genome. And this is shown here. So in the blue bars show integration sites of T cells after infusing, after transplantation into this patient of the stem cells, and T cells before infusion. This means that the LTRs of the retrovirus, or the integrated retroviruses, influence expression also of the gene next to where they're integrated. And because the distance to the transcriptional start site is so close, of course you can expect that expression of that gene is somehow mixed up. So how can we improve the architecture of these retroviral vectors to improve the safety profile? So what one can use is switch from gamma retroviral vectors to lentiviral vectors. What do we know? The lentiviral vectors have a tendency to integrate within transcriptional units into the genes. It's somewhat, that's what we know in the meanwhile, a somewhat reduced genotoxicity compared to the gamma retroviral vectors, but nevertheless they integrate more or less randomly into the genome and also can mix up or can perturb the expression of the gene where it integrates into. We can use self-inactivating vectors. I mentioned that before. So you take out the U3 region of the LTR so that the LTR, the enhancer promoter region of the virus itself, is dead, which is a very, very strong enhancer. Take it out, and the chance to disturb the expression of neighboring genes is much, much reduced. Instead, if you use an internal promoter, which is tissue-specific, for example, which has a different strength, much lower than the endogenous LTR of the virus, you get an increased safety profile. And you can try to just hit or transduce those cells you're interested in, you want to treat, by pseudotyping of the vector particle. So again, how do you produce a self-inactivating vector? You delete the U3 region and end up with a syn self-inactivating LTR. By using an internal promoter, which can be tissue-specific, you again increase the safety, so you get the gene only expressed in cells you want it to be expressed and not in all of the cells. You can do pseudotyping, for example, by using the glycoprotein of totally different viruses, like the vesicular stomatitis virus, like the lymphocytic choriomeningitis virus. All these viruses have a different tropism. They infect different cells. So if you use the glycoprotein of those viruses, you redirect your vector to the cell type you want to transduce. And this is depicted here. So now, switching gears a little bit, we can now use those lentiviral or retroviral vectors. Retroviral vectors are such for treating malignancies. And one of those, which everybody has heard about in the meanwhile, are so-called CAR T treatments. So CAR stands for chimeric antigen receptor, meaning that you have a patient having a malignancy, a lymphoma, a leukemia. You take out the T cells, you take out lymphocytes or immune cells from that patient by leukapheresis, which is a type of a centrifugation protocol. Use those cells to transduce them with a retroviral vector expressing this chimeric antigen receptor, which is directed against a surface protein, or a protein which is, in the best case, only expressed by the malignant cell. So that you redirect the immune cell to attack the tumor, lymphoma. And that, if it's a T cell, is then called a CAR T cell, again, chimeric antigen receptor T cell, which you reinfuse in the patient. That's a very, very successful treatment in this like exploding at the moment. These CAR Ts or CARs have evolved over time. It's not so important now here to memorize which generation of CARs there are with different signaling, intracellular signaling domains. But there are different flavors and colors of these CARs. You can also modify, of course, not only T cells, but, for example, natural killer cells. A lot of research is ongoing, and also in the meanwhile, clinical studies using different immune cells with all their advantages and disadvantages were out there. Trying to improve also the activity of those cells, the tumor killing activity, and the safety, of course. So these are autologous treatments. You take cells from a patient, manipulate the cells, give it back to the patient, which is a lot of work and very costly. So these treatments are expensive. What you would like to have is, again, something which you could use off the shelf, which you can produce in great amounts and from few donors, and then use for many, many patients. And one idea would be to take a donor, for example, T cells from whatever the donor is, a normal healthy donor, insert the chimeric antigen receptor by, for example, retroviral transfer, or by other methods, by homologous recombination, and then remove the endogenous T cell receptor. So those cells commonly used for as CAR Ts are manipulated, not only express the CAR, but also still the endogenous T cell receptor, which also recognize a target. So to focus the activity of the T cells so that it only recognizes one target, you would want to remove the endogenous T cell receptor, and you could, for example, remove a molecule which is necessary for allogeneic activity, called CD52 or cluster of differentiation 52, just as an example, to reduce or minimize the alloreactivity leading to rejection of the recipient. By that, producing a universal CAR T cell which can be used for any patient is one idea. The other idea, again, is to use induced pluripotent stem cells as a start. So reprogram cells from someone, produce induced pluripotent stem cells. We would do gene editing, for example, removal of the endogenous T cell receptor alpha beta chain, introduce the CAR construct, and either make hematopoietic stem cells or CAR T or CAR NK cells, which you can use to treat the patient having a cancer or leukemia, lymphoma, whatever. So this would have the advantage that, again, you can have few donors, generate a bank of different CARs, for example, CAR expressing progenitors, which you then could use to treat many, many recipients, reducing the costs of production of these cells, making it affordable for the society to have to treat as many patients as possible and not having such very, very high costs, which we have at the moment. And we're heavily moving into that direction. So a Japanese company, for example, and a university in Kyoto are working on iPS cells, which they have already differentiated towards T cells, which were manipulated to become CAR Ts, derived from iPS cells, so then iCAR T cells. Again, as I said before, with the goal to have high scalability, lower cost of production, and something which you can use off the shelf to treat cancer for as many patients as possible. And not only, as you can imagine, in Japan, these things are ongoing, but also in other areas in the world, also in the US, of course. And the company Fate Therapeutics, just to mention one, is at the stage where they already use it in clinical trials, which was reported a few years ago at the Aachen meeting. So we're making huge leaps towards the use of iPS-derived CAR T cells to make them available for many, many patients. And with that, I'm at the end of this lecture. Thanks for your attention. Thanks for listening to me. I hope you learned something and can profit from that. Thanks.